Okay, now we're moving on to the stomach in section 6. When we're talking about the stomach, its primary function is just to really act as a temporary food storage bank until we can get the food contents into our small intestine and large intestine. We can only secrete small amounts of the contents of our stomach into the small intestine, which we'll talk about later. So we hold most of the food in our stomach and release small amounts into our small intestine. We also begin protein digestion here. If you remember back to our mouth with salivary amylase, we began carbohydrate digestion with salivary amylase, but we really didn't start breaking down any of the other macromolecules. So the stomach is our first organ where we can actually begin breaking down proteins. So what's entering in from the esophagus into the stomach is that bolus, that compact food ball that's mixed with saliva. But in our stomach, it's going to go through both physical and chemical process to help break down the bolus into something that we call chyme, so more paste-like. If we don't have any food contents in our stomach volume is about 50 milliliters, and we can see these ridges in the mucosa of the stomach itself, and these ridges are called rugae. Once we start filling our contents, the rugae are going to disappear, and we can actually hold up to about 4 liters to 1 gallon of food in our stomach and stretch it to that capacity. So we mentioned earlier that there's 4 general layers or tunics to the walls of all gastrointestinal organs. However, some of these walls or layers or tunics are modified. So we have 2 that are modified in our stomach. One is the muscularis externa. So the muscularis externa normally consists of just the circular smooth muscle layer and the longitudinal smooth muscle layer, but in our stomach we have an additional layer, and the additional layer is the oblique layer. And the purpose of having a third muscular layer is to really help increase the physical breakdown of the contents of the stomach. And also, again, we have to get the contents of our stomach into our small intestine, so it also helps assist in that. The other tunic that is modified is our mucosal layer. We have predominantly simple columnar epithelium with mucus cells, but we also have some additional cells that we're going to talk about a little bit that make up the gastric pits and gastric glands and help produce the gastric juice needed for further chemical breakdown. So let's start talking about the cells that we can find in the mucosa. Over here, this is what I was talking about earlier. This is our gastric pit, and that's going to lead and develop into the gastric gland. We have 4 types of cells that can be found in the gastric pits and glands collectively. So if we're talking about the mucous neck cells, they're at the neck of the gastric gland, and their function is to secrete an acidic mucus. Unknown function. However, anytime you have something that's acidic, it contributes to help inhibiting bacterial growth. So that's the most likely function of this acidic mucus. The other important cell and its secretions are the parietal cells. So the parietal cells are shown in blue. They're kind of interspersed throughout. And they have 2 important secretions, one of which is hydrochloric acid. So because it's an acid, we would expect that its secretions involve an extremely acidic or low pH substance, so around a pH of 1.5 to 3.5. The functions of secreting the low pH involve helping to denature proteins. Again, we're trying to break down proteins, so we need to denature them first. It's also going to be crucial in activating pepsin. So we need pepsin is our enzyme that's responsible for protein breakdown or degradation. Also helps break down plant cell walls. And again, anytime we're talking about something that's acidic, that's going to help inhibit bacterial growth. The other important secretion from the parietal cell is the intrinsic factor. And the intrinsic factor is really important because it's required in order for us to absorb vitamin B12 across our intestine. And we'll talk about why we really need to have vitamin B12 being absorbed. The third type of gland cell is going to be our chief cell. So here's our parietal cell over here in blue. We're zooming in a little bit further. It's our pitchfork looking one that secretes hydrochloric acid as well as the intrinsic factor. And then our chief cell. Our chief cell is responsible for secreting pepsinogen. Pepsinogen is the inactive form of the enzyme that's responsible for breaking down proteins. So it's the inactive form of pepsin. What we need is hydrochloric acid in order to activate pepsinogen into its active form, which is pepsin. Another important secretion from the chief cell is lipases. So lipases are enzymes that digest lipids or fats. So not a huge proportion of lipids are being digested in our stomach, as well as when we talked about our mouth, because we do have salivary lipases as well. Not the bulk of the lipid breakdown, but we can digest about 15% of our lipid intake in our stomach as well via the lipases secreted by the chief cells. The fourth type of cell we can find is our enteroendocrine cell. So those are our green cells in this picture. So these cells are a form of endocrine cells, so they can secrete hormones. They secrete chemical messengers. Some of these chemical messengers can act as paracrines, meaning they are acting on either the same cell or a very close cell. That would include serotonin and histamine. And then we also have our hormones, which are going to go and act farther away. And that includes somatostatin, which can also act like a paracrine, as well as gastrin. So we are going to incorporate some more hormones here. The one thing that I kind of want to point out, these are the ones that you're responsible for, the ones that I have in my slides, even though your textbook may mention more. What you're focused on here is the hormones that are produced by the stomach itself. And we also later on are going to have hormones that are produced by the small intestine, specifically the duodenum of the small intestine. Now, just because they're produced by the stomach or the duodenum doesn't mean that they act where they're produced. A lot of these will travel and go and act somewhere else. So this is just where they're produced, kind of understand, is it the stomach that's producing them or is it the duodenum that's producing them? But where do these hormones or paracrines go to actually act? Okay, a little more on the mucosa. The stomach mucosa is exposed to really, really harsh conditions because of the high acidity. So we have a high hydrogen ion concentration in the stomach, almost 100,000 times higher than we find in our blood. We also have the protein digesting enzymes that can be found as well, and we don't really want our protein digesting enzymes to actually start digesting the stomach itself. So we have to protect the stomach from all of these different things. So how do we protect the stomach from getting digested from the contents that the stomach cells are actually secreting? One is that the stomach is protected by there being a thick coating of a bicarbonate-rich mucus on the stomach wall. So the bicarbonate-rich mucus, basically, we'll talk later on about the bicarbonate ion. It can act as an acid or a base, typically. So if we have too high acidity, it can bind up the excess hydrogen ions. Another way it helps protect against itself is the epithelial cells of the mucosa. They're joined by tight junctions. So a tight junction's analogous to like a Ziploc bag. Nothing can really get in and nothing can really get out, so that we're preventing gastric juice from leaking into the underlying tissue layers. Another protection mechanism is that any of the damaged epithelial mucosal cells, they're going to be shed very, very quickly and replaced by underlying cells. So there are stem cells in the gastric pits, and once we start damaging the external mucosal cells, the ones that we can produce from those stem cells underneath will get pushed forward. So we actually replace all of our epithelial mucus cells about every 3 to 6 days because we're constantly shedding them. Just to bring in some clinical aspect to this chapter, gastritis, what is that? We're talking about inflammation. So inflammation caused by anything that breaches the stomach mucosal barrier. So we also have peptic or gastric ulcers, and anytime you have ulcers, that's basically some sort of erosion in the stomach wall. Most ulcers, they found out not that long ago, fairly recently, I'd say within the last 20 years, are actually caused by a bacteria. And that bacteria is Helicobacter pylori or H. pylori, and that's what's in green over here. And Helicobacter pylori is actually like a corkscrew shaped bacteria, and what it does is it kind of goes in and buries into the mucosal wall of the stomach, damaging the barrier and exposing it to the high acidity, leading to the inflammation in the mucosal lining. Okay, digestive processes of the stomach. There's several digestive processes. We already talked about it can carry out the breakdown of food, both mechanical and chemical breakdown. It primarily functions as just acting as a temporary storage bank for holding the food until we can deliver small amounts of that chyme into the small intestines. We do begin breaking down one of our macromolecules, specifically our proteins, both hydrochloric acid that helps denature it and activate the enzyme we need for protein digestion, which is pepsin. Some lipids can also be broken down here from the lipases that are secreted by the stomach. And if we're talking about alcohol or some drugs like aspirin, those can also be absorbed into the blood from our stomach. Really, like I said, primary function of the stomach is to act as a food storage bank and to deliver small amounts of food into the small intestine. Really, the only essential function of the stomach is that secretion of the intrinsic factor, because we have to have intrinsic factor in order to absorb vitamin B12 across our intestinal wall. Why do we crucially need vitamin B12? Well, in order for our red blood cells to mature, we need vitamin B12. If we don't have intrinsic factor and we can't absorb vitamin B12, that can lead to pernicious anemia. So anytime you have pernicious anemia, all you need to do in order to treat it is to get B12 injections. So now we can talk a little bit about the regulation of gastric secretions. Typically, our gastric mucosa will pour out as much as 3 liters of gastric juice. This is a huge amount. We have both neural and hormonal mechanisms that help control gastric secretion. If we're talking about the neural mechanisms, it's going to be our vagus nerve. When our vagus nerve is stimulated, that's going to increase secretion, leading to acetylcholine being released. Acetylcholine release is going to stimulate the output of gastric juice. Our sympathetic division is going to cause a decrease in acetylcholine secretion. Hormonal mechanisms of gastric secretions is predominantly controlled by gastrin, and it's going to stimulate the secretion of the hydrochloric acid by those parietal cells in the stomach. Also can help with hormones being secreted from our small intestines. So really, when we're talking about control of the hydrochloric acid secretion by the parietal cells, 3 different things can control it. Gastrin, which is our hormonal mechanism, acetylcholine, which we talked about being released if we have vagus nerve stimulation, and histamine, which I haven't mentioned yet. But all 3 of those control how much hydrochloric acid is being secreted by the stomach. So when we talk about gastric secretions, they're broken down into 3 different phases. We have the cephalic phase, the gastric phase, and the intestinal phase. So the cephalic or the reflex phase of gastric secretions is going to occur before food even enters the stomach. Typically, it's only a couple minutes long, and we would trigger this phase by smelling something, tasting something, the sight of something, or even just thinking about something. Any one of these triggers can act via that vagus nerve to stimulate gastric glands to start with the digestion itself. If we're talking about this phase, we can also have inhibition. So loss of appetite would inhibit this reflex stage, or even depression, ultimately leading to a decreased parasympathetic activity. So the second phase is our gastric phase. This lasts a lot longer. Basically what happens here is once the food reaches our stomach, both the local neural and the hormonal mechanisms are going to help initiate this gastric phase. And this lasts about 3 to 4 hours and provides the bulk of the gastric juice being released, so about 2/3 of that gastric juice. When we're talking about stimulation, the most important stimuli are going to be something called distensin, as well as peptides and low acidity. Because we would want this phase, we would know we have contents in our stomach because we have low acidity from a hydrochloric acid secretion. We have the presence of proteins in our stomach, and we can sense that our stomach is stretched a little bit. The chemical stimuli, again, like I mentioned, so things like partially digested proteins, caffeine, and the low acidity, those will all activate the G cells to secrete gastrin. Once we release gastrin, gastrin's going to initiate hydrochloric acid release from the parietal cells, and we know that hydrochloric acid is going to help activate pepsinogen into pepsin. Binding of gastrin to receptors on parietal cells also helps stimulate enteroendocrine cells to release histamine. So all of these is kind of like a positive feedback cycle. All of these are going to enhance the breakdown and release of hydrochloric acid. So we do have buffering action of the ingested proteins, causes the pH to rise, which activates more gastrin secretion, and it's kind of this cycle that's going on. Inhibition of the gastric phase, what would cause this? So low pH inhibits gastrin secretions, stress, and fear. Typically, we would inhibit this phase between meals if we're not eating, and it occurs during digestion as a negative feedback mechanism. So the more protein that we have coming in, the more hydrochloric acid we have being secreted, which means we're going to be lowering the pH anymore. So the low pH is going to cause an inhibition of gastric secretion. Okay, the intestinal phase of gastric secretion begins with a brief stimulatory component followed by inhibition. So when we're talking about the initial stimulatory part of the intestinal phase, that's when we're having partially digested foods entering into the small intestine. This causes a brief release of intestinal gastrin. Intestinal gastrin is also called enteric gastrin. And this hormone is going to help encourage the gastric glands to continue their secretory activity. And this is a stimulatory effect. It's very, very brief because it's overridden by inhibitory stimuli as we continue to fill the intestines. So next step would be inhibition of the intestinal phase, and we have 4 main factors in the duodenum to cause it to stop the gastric secretion. So the first one is distension of the duodenum. So we basically stretched it as the chyme is coming in from the stomach. Also, things like the presence of acidic, fatty, or hypertonic chyme all trigger both neuronal and hormonal signals to tell the stomach we have enough food and enough chyme coming in already. So this is, again, mechanisms in which we stop this. These 4 same factors also decrease gastric emptying. So they're going to kind of halt or stop the gastric activity to help protect the small intestine from receiving too much acidic contents at once. So we can inhibition is achieved in two different ways. We have the enterogastric reflex and enterogastrones. So the enterogastric reflex, basically what that's saying is that the duodenum, so the first segment of the small intestine, inhibits acidic accretion in the stomach by short reflexes through that enteric nervous system that we talked about, as well as by long reflexes that are involving the sympathetic and vagus nerves. When we're talking about enterogastrones, these hormones are released by those enteroendocrine cells and then duodenal mucosal epithelium. And we have two important ones. One is secretin, and the other one is cholecystokinin, or CCK. And these hormones are going to inhibit gastric secretion, and they also play some other roles that are pretty important. So here's a nice little table, kind of what's going on, things that can would stimulate gastric secretion and other ones that would inhibit it over here, and comparing that to the three different phases that we've talked about. So I would kind of go through this chart and make sure you understand what's going on with these different phases for gastric secretions. So here's the table on the hormones. Again, there's way more hormones on this table than you're responsible for. I believe I highlighted all the important hormones in blue bold font. Those are the only ones I will hold you responsible for on this upcoming exam. The rest of them, you are not going to be responsible for. Okay, mechanism of hydrochloric acid formation. So our parietal cells, when they're stimulated, are going to pump hydrogen ions from carbonic acid breakdown, which we'll talk about, into the stomach via a hydrogen potassium ATPase protein pump. So the parietal cells are pumping hydrogen ions into the stomach lumen, and we'll talk about how we get the source of hydrogen ions that is pumping. So once we have hydrogen ions pumped into the stomach lumen, we have the bicarbonate ion exported back to the blood. How do we export the bicarbonate ion back to the blood? Via a chloride shift, essentially, kind of like what we saw in red blood cells. So we have a chloride being exchanged for a bicarbonate ion. So anytime we have ions moving in the opposite direction, we call the channel they're moving through an antiporter because they're going in opposite directions. A symporter would mean that the ions are moving in the same direction. So as we're doing this, we get an increase of bicarbonate in the blood, leaving the stomach, and this is referred to as the alkaline tide. So the chloride ion is going to be pumped out to the lumen to join the hydrogen ion, and we're going to ultimately form hydrochloric acid. So this is an image kind of illustrating what's going on. We're focusing in on our pitchfork parietal cell because that's the one responsible for hydrochloric acid secretion. So step one, we're talking about the hydrogen ion and the bicarbonate ion are being generated from the dissociation of carbonic acid. So we've seen this before. So because here's our bicarbonate, carbonic acid is really, really unstable. So it will almost immediately dissociate into a bicarbonate ion here and a hydrogen ion here. So once we have that being produced from carbon dioxide and water that are entering into the parietal cell, we have a really important pump called the hydrogen potassium ATPase pump that pumps that hydrogen ion from that breakdown into the lumen. So this is our stomach lumen over here in orange, and potassium into the cell itself. The potassium returns to the lumen through the membrane channels, so we can see it being returned back through. The bicarbonate ion leaves the cell. When we have the bicarbonate ion leave the cell, here's the bicarbonate ion, leaves the cell into the interstitial fluid and ultimately enters our bloodstream. This is referred to as the alkaline tide, Anytime we are losing a negative charge, we have to bring in a negative charge in order to keep our electrical balances the same. So every time we lose a bicarbonate ion to our interstitial fluid and blood, we have to bring in a chloride ion. And we saw that before in our red blood cell. The chloride ion enters into the parietal cell, is going to enter out into our stomach lumen, where it combines with the hydrogen ion that we generated from the breakdown of carbonic acid. So gastric contractile activity, we have those peristaltic waves move toward the pylorus. We have them moving, or the waves moving, at a rate of about 3 per minute. We have the basic electrical rhythm, also called BER, and it's going to be set by enteric pacemaker cells. So anytime we have an organ that's contracting that we don't have voluntary control over, so it's involuntary, like we saw with our heart, what's initiating contractions are always going to be pacemaker cells. And the pacemaker cells are going to be linked by gap junctions. And anytime you have a gap junction, the purpose of that junction is for communication, so that we can communicate with all the smooth muscle cells in that entire muscularis, so we can contract it as a unit. We have two important things that influence the force of contraction. One is distension, and the other one is gastrin, which we've talked about. And we produce about 30 milliliters of chyme. However, we can't, like I told you before, we can't just shove all the chyme that we're producing in our stomach into our small intestine at once. Instead, we only want very small amounts to actually enter into the duodenum at a time. So only about 3 milliliters of chyme is being delivered into the duodenum, or less, of the small intestines. The rest, so say if it was 30 milliliters of chyme, the rest, the 27 milliliters, would be propelled backwards into the stomach, and it would continue to mix. So it's basically this process where it's moving the contents toward the duodenum, squeezing out a little bit, 3 milliliters, then contracting and squeezing it backwards to the back end of the stomach, and then back towards the duodenum, squeezing out a little more, and basically mixing and keeping going back and forth, pumping action, until it gets all of the contents of the stomach into the duodenum, or the small intestines. So this is kind of what I was telling you just now. We have propulsion where we're using the peristaltic waves to move from the fundus towards that pylorus. This is our pylorus over here. And as we do that, we move it towards that pylorus over here. We can allow about 3 milliliters to go through the pyloric valve and into the duodenum here. Once we've allowed some of the chyme to go into the duodenum, we're going to contract again, and we're going to squeeze the contents back up towards the fundus of the stomach, and then starting back over at 1, where we stick it back down towards the pylorus, squeeze a little bit out into the duodenum, contract, bring the contents back up to the fundus, and so on and so forth, till we basically empty the contents of all of the contents of our stomach into our duodenum. So how do we regulate it? The stomach usually empties completely within a couple hours after a meal. If you have a larger meal, that means you have greater stomach distension, so you've stretched your stomach more, and it's going to cause your stomach to want to empty faster. So we're talking about control of emptying into the duodenum, or the small intestine. So we have receptors in our duodenum called duodenal receptors that respond to stretch and chemical signals. And we have the enterogastric reflex and our enterogastrones that act to help inhibit gastric secretion and therefore duodenal filling. So like I mentioned, and within a couple of hours, we can empty our stomach. But if we increase the fatty chyme that's entering into the duodenum, meaning the higher the fatty content in our stomach, that's going to increase the time to about 6 hours to completely empty from our stomach. So, and then if we're talking about carbohydrates, those are going to move quicker from our stomach into our small intestine. So if you've ever heard, like if you eat a lot of fats or even proteins, that will help keep you full longer, it's actually true because you're moving the contents of your stomach, the chyme, much slower with the higher lipid and higher protein ratio you have making up the chyme. The opposite is true for carbohydrates. Carbohydrates help the chyme enter much quicker and move quicker into the duodenum. So your stomach is emptying faster with the higher carbohydrate content that's in there.